I finished textile faculty at the Lodz University of Technology in 2000. In 2004, I finished my PhD, which was strongly chemical. In 2012, I finished my habilitation, which, in contrary, was rather, I would say, concerned on textiles. Now I'm a head of physical chemistry of polymers division in the large department. Okay. My scientific interest concerns on surface modification of textiles, on thermal analysis of different materials, and generally on preparation of different polymeric components of different polymeric materials. My university is on the 4th place in the rank of the Polish technical universities. We are concerning strongly on the internationalization, so I would like to invite you to, maybe when you finish your study here, to go for the 3rd step study at my university. We have international school of PhD students, so you can come when you finish 5 years of study, so you have the master title, you can go, you can apply, and you can come to our university for PhD study. Some parts of our university are in old buildings like this one, in old villas, some in modern buildings built exactly for that university. This university was initialized in 1945, initially with 3 faculties. Now we have 9 faculties, 70 institutes and departments, and 120 specializations. You can study, if you prefer, you can study in English, you have some possibility to study in French, but mostly, of course, you have to study in Polish. But we have the International Faculty of Engineering, which is open for everybody. Now we have about 20,000 of students and about 3,000 staff members. Very modern, fully computerized library. You're going to the library and you almost cannot see any stuff. Everything is mechanical. We have even our own radio station. I would like to tell you a couple words about a couple definitions, because I would like to be well understood. So what I mean about when I will talk about biodegradation. Biodegradation is a conversion of the substance to different neutral substances like CO2, H2O, and the biomass by a regular biochemical process. What I mean as bioactive. Bioactive is a material that exhibits some positive effect on living organisms, on living systems. And bio-based is a compound which is usually derived biological products from biomass. Decomposition, it's a process of destruction of the material, and utilization is a changing of wastes into harmless destruction products. And another slide about definition. Biomacromolecule, it's a molecule produced by living organisms. Biomass, it's an ensemble of organic substances that constitute living systems, and of course its post-mortem residues. Biopolymer is a substance composed from one of the biomacromolecules. And biomedical, it's a material which can have practical activities related to any biological or medical therapy. Humankind, when we look at the history of humanity from the beginning, from the historical point of view, humankind necessities are, we have some basic necessities. The first is to be safe, to have some food, to live with some group, but we are not thinking about it, that to having clothes is one of the basic necessities which the humankind can have. So we always were using some kinds of clothes. At the beginning, it was animal skin. After that, shortly after that, we were using wovens. Next were knitted fabrics, and finally, we used chemical fibers. The basic source of natural fibers are cotton, made from cotton plant, wool, made by ships, silk, made by silkworms, and all natural fibers. Even in ancient Greece, people were using wovens, and these wovens were made from wool and linen. But even in the times before Christ, it appeared wovens which were made from cotton, and this kind of wovens were imported from Middle East. We can divide textile raw materials into two groups, natural as a natural and a chemical. Natural made by plants and animals like cotton, wool, and silk, and some chemical, which can be organic and anorganic. This which are colored here in green are biodegradable. This which are shown here on red are not biodegradable. This is only one group in chemical part which can be treated as a biodegradable polymer material. They are made from regenerated cellulose, for example. And we are looking for some solution how we can make biodegradable polymers using chemical ways. Biodegradable polymers are on the topic on our interest now. Why? There is a couple reasons that there is still increasing interest in using biodegradable materials. Of course, the basic reason is that they can be easily utilized. So the number of people in the world is still increasing. We are using the comfort of living is going up. We are using a lot of not only clothes. Clothes is not the main problem, but we are using a lot of different materials. Mostly, I mean, disposable materials. And we are producing a lot of wastes, and we don't know what to do with these wastes. So using biodegradable polymers is kind of solution of that problem, because we can produce more materials and it will be degraded in the natural way. This is the one of the reasons. Of course, access to the resources is a second reason, because there is not enough natural sources for everyone in the world. It's too many people in the world. So we have to use chemical fibers. There is no other way now. And additionally, chemical materials and chemical fibers are cheaper than natural fibers. The next reason, they have very good mechanical properties, so its fastness is much better. And we can produce chemical fibers in such a way which we want. So we can have such materials which we need exactly. We can customize these products. So we can make such fibers which are necessary for our aims. First, very popular and produced on a large scale chemical fibers were polyamides and polyesters. They have very good durability. Later was polypropylene fibers, and this polypropylene is additionally very cheap. But the problem with regular chemical fibers is such that they are not biodegradable. They are responsible for microorganisms, and they cannot be in regular process biodegradable in the nature. So people are looking for different solutions, and this is, for example, some kind of solution to produce viscose fibers. People are making viscose fibers from regenerated cellulose. Cellulose is taken, for example, from trees or somewhere. But this is not a good process, mostly because it's very difficult to find some solvent for cellulose. And you have to use complicated, as you can see here, a complicated chemical process. And finally, together you're producing a lot of toxic gases. It is dangerous. And now viscose fibers are not very popular, I would say. And as I said, the clothes are not the main problem. The main problem are disposable textiles. For example, for hygienic reasons, we are using a lot of textile materials. They are not clothes, but I would say textile materials, which produce huge amount of wastes. And even if we want to come back to using only biodegradable natural fibers, it is not possible. For example, in 2005, people globally used about 52% using fibers which were used by people in the world were chemical fibers. 92% from this 52, were synthetic fibers, so I mean 8 from regenerated polymers. We have different methods for production of chemical fibers. We always try to use, if it is possible, we always try to use melting technique because this is the most comfortable. We are not using solvents. The technological process is very easy. So always when it is possible, we prefer melting technique. Why chemical fibers are not biodegradable? Because they are responsible, they are resistant to microorganisms. And this, from one point of view, it can be good because we want to use textiles which are resistant to microorganisms, but on the other hand, such material cannot be biodegradable in a regular way. How long we have to wait for biodegradation of different kinds of materials? And here, I will read it because it is too small to see probably. So paper towel will the biodegradation of paper towel will take from 2 to 4 weeks. Newspaper will degrade in 6 weeks, cardboard box in 2 months, waxed milk carton only 3 months, apple core 2 months, cotton gloves from 1 to 5 months. Of course, these times are strongly dependent on the conditions, so you have to take into account humidity and temperature. You can have some conditions in which the material will not be biodegradable. So this data are for some special conditions. Plastic beverage holder, 400 years. Plastic bags, 100 years. A glass bottle, we are not sure. Disposable diapers. We are producing a lot of disposable diapers. We have, this is very comfortable in use. And the time of biodegradation is between 50 and 100 years. This is a mixture. The final product here is a mixture of organic and not organic material, and the biodegradation is very difficult. And for example, next example is monofilament fishing line will degrade in 600 years. So we can only estimate this time because we are not living in the world too long. We are not using this material so long to be sure, but we are estimating this time for 600 years. But we know some thermoplastic polymers, I mean people, some thermoplastic polymers, so thermoplastic which can be processed in melting technique, using melting technique, which are completely biodegradable in environmental behavior. And these polymers are aliphatic polyesters made from such monomers as glycolic acid, lactic acid, butyric acid, or caprolactone. You can see the structures here. And there is a large market for such materials. We want to use such materials in different applications. For example, as a surgical treats, not for the skin, but when you have an operation and it is necessary to use surgical treats inside of our body, it is always good to use such material which can be really biodegradable, and some kinds of implants which should not to be in our organism too long, only for some time. The advantage of using, for example, polylactide is this, that we can, the time of biodegradation is different in different conditions, and we can estimate the time. So, for example, we can produce such polylactide, which will be biodegradable in such form, it will be biodegradable in 6 months. It is enough for our organism to build different structure close to this material, and it will be degraded to lactic acid. And lactic acid is produced by our body, and it is very good for the implantation process. Why I'm talking about the polylactide? Because we were working on this material couple years using Biogratex project. In this project, we developed some new products made mostly from polylactide, which we tried to apply it in agriculture, in filtration aims, for hygienic materials, and for some medical aims. For agriculture, for example, when you have some plants, you need to cover these plants with some materials, and mostly it is so that you have to take the used material when it is not necessary to stay, but polylactide you do not need to take because it will be biodegradable after that time. Firstly, first commercial producer of polylactide was Carters in 1932. So thanks to this man, we can make polylactide materials today. Generally, from the chemical point of view, we have several ways, several routes for production. 2 main monomers which we are using are lactic acid and just lactide. I will show you. So plants using photosynthesis process are producing carbohydrates. Bacteria in the fermentation process can produce lactic acid, and next we are using some catalysts, and in the polymerization or polycondensation process we can have finally polylactide. Melting temperature of that polymer is 180 degrees, glass temperature about 55 degrees, and the cost 1 kilogram is about €2. So it is not very cheap, but it is not expensive. I mean the cost in the industrial process, which we estimate for €2 per 1 kilogram. The most popular way, the most common way for polylactic production is the ring opening polymerization of lactide, and mostly it is necessary to use different catalysts. Typically it is thin outstate. It can be made in solution, in melt, and as a suspension. about the project I was talking to you. We were looking for different producers, and when we want to compare the properties, because so we can have we can produce in our laboratory polylactide material, but we would like to have this biodegradable material to have similar properties as different chemical polymers, and we can compare. Tensile strength in megapascals for polylactide is 570, so it's slightly smaller than polyethylene, polypropylene, and polyethylene terephthalate, but it's in the same range, I would say. Elongation break 30% between polypropylene and polyethylene terephthalate, and Young modulus 6 close to polyethylene. So it's not bad. Maybe in some aspects it will be not enough, but generally we have to admit that we can produce polylactide with quite good mechanical properties. From the textile point of view, we can produce polylactide in the form of fibers, like here you can see, and we can produce direct, non-woven, like here. So using melting technique, in one-step process from the polylactide, we can change this polylactide into the textile product. Mostly you have to use couple stages process, but this time it is easy to produce non-woven in one-step process, so it is real advantage. We can control, we can change the properties of the material using mixture of polylactide L and polylactide D. For example, the melting temperature of polylactide L can be increased even up to 50 degrees, and the heat deflection temperature can be increased from 60 degrees up to even 190 degrees by physically blending this L polymer with polymer in the form D. Now on the market we can find materials which were made from polylactide. As I said, surgery treats from about 1960. Some microspheres for different applications, nanofibers. And now it is easy to see this is this fiber in comparison to human hair. So very thin fibers, and capsules with some medicines inside. They're the main medical applications, but of course, for me, the most interesting form is the form of fiber, and in this form, we can use polylactide now, or using polylactides, for example, as upholstery, disposable garments, like here, awnings, feminine hygiene products, and of course, diapers. This is very important. This is a very important application. Okay, thanks to this easy degradation into innocuous lactic acid, polylactic is used very often for medical implants in the form of screws, pin, rods, and even as a mesh for hernia, against hernia. You can have it inside your body in a safe way. This, thanks to its low price, sometimes we are using this for some packaging materials, cups. We found cups made from polylactide, and bags also made from this material. You can, this polylactide is in the form of film. It shrinks upon heating, so it allows us to use this in shrink tunnels. And it is useful for producing of loose film packaging, compost bags, food packaging, and disposable tableware. And even shirts are made from polylactide, microwavable trays, hot fill applications, and some engineering plastics even. Why polylactide is biodegradable? The classical, I would say, mechanism of biodegradation is using enzymes, which are catalysator for two processes like oxidation and hydrolysis reactions. And as an effect of using of these processes is cutting the main macromolecule on smaller molecules. And this is important to have good quality polymer, because when you have no more additives and contaminations, it will be degraded. It is the first step is a polymerization, and the degradation can be mostly in the very easy depolymerization process into the monomer again. The monomer is fully safe for using by human. It is important to control the conditions of biodegradations, because if we want, as I said at the beginning, if we want, we can use polylactide for many years without any changes of its structure. Everything depends on the conditions. So when the temperature, we have to control the temperature and the humidity. For example, in active compost, the biodegradation is very fast, but without high humidity and without high temperature, we can use it for many years and still we can have the same properties. But in proper conditions, it can take only a couple of weeks, the biodegradation process. So firstly, when we talk about the biodegradation, about the hydrolysis process, first macromolecules are going to oligomers, and next is chemical or enzymatic hydrolysis, and large macromolecule is cutting the hydrogenation process, and macromolecule is cutting into the small parts, and finally we have monomers. The mechanism is also not complicated. Firstly, we have ester bond decomposition, next nucleophilic attack, and finally acidic catalysis, leading us to biodegradation. But the main part of this lecture is to show you how we can change the properties of that material. So the producing of polylactides is known, as you saw, since many years. But we would like to change the aim of that project, which I showed you, was, because it finished this year, this project, was to change the properties. And it was one group which was working on using some additives into the melting to change volumetric properties, but my group was working on the changing of surface properties using different processes, different techniques. The first possibility is to make a grafting process. So this is quite complicated process. You know, probably the grafting, you have the surface of one polymer. This is polylactide, and you can graft. So you can graft on the surface different polymers. You can have different macromolecules here. It is possible to have grafting from and grafting to. So the mechanism is that you have the polylactide. You can make some activation process to have some active groups on the surface. It is not easy. I will show you in the next slides. And finally, we are using monomers here. And it is important to start polymerization process of these monomers. And we want to attach the end of this macromolecule to the surface, to the activated surface of the polylactide material. And what we can have thanks to this process. The main advantage of this process is that we can have completely different surface properties. Surface properties in such case, in comparison to the beginning, is dependent only on this polymer. So the mechanical, the volumetric properties, we have still the same. It is still polylactide, and it is still biodegradable, but the surface properties are completely different. This process is very efficient, and but it's not easy, it's not cheap, but it allows us to change the surface properties in permanent way. So this is a strong modification. At the first slide, I show you how we can attach polyacrylic acid to the surface using a grafting process. Ammonium persulfate was used in these processes for initialization. We tested, we controlled different times for using ammonium persulfate, different concentrations of ammonium persulfate, different temperature, different processes, different conditions of this process, different concentrations of acrylic acid, and we found the most efficient way for production of grafted material, and which it is important to find the way how we can control the structure, because to make modification is one step. We have very thin, after this modification, we have very thin layer on the surface, and it is difficult to have technique, to find technique which can be used for controlling of this process, because the regular spectroscopic techniques are not cannot be used here because the layer is too thin in comparison to the main material. And only I would like to show you here that we have to use temperatures close to 80, and it takes 1-2 hours. So it is not, as I said, it is not easy process because it's 60 degrees. The number of acidic groups on the surface was too small for us. And how we can control the structure. For example, here there is FTIR 80-year spectrum. So the regular FTIR is not enough. We cannot see during the transmission technique. But we have to use some surface techniques. For example, here is ATR. So it gives us information only about the surface. Here you can see the A, it's an initial polylactide fiber, B, it's a grafted fiber, and C, it's a polyacrylic acid. And you can see that we have here a strong signal connected with carbonyl group from acidic. I know that probably at the end of the room it's not easy to see, but believe me, that here we can see the signal connected with carbonyl group from the polyacrylic acid. So FTIR spectroscopy can be good technique if you have such device. It can be good technique for control of the grafting process. Next technique which I would like to show you is entrapment process. The idea of this technique that we are using some mixture of solvent and non-solvent. This mixture is changing the surface, and when close to the surface are different macromolecules, ends of that macromolecules can be attached to the changed surface. It can be swelling process. And after that, we have permanently combined some different macromolecules, and it can be also efficient way for functionalization of polylactide surface. Maybe it's not so strong connection, maybe it is not so permanent like by grafting process, but it is easier to do. It is easier to prepare. As a mixture of non-solvent and solvent, we use ethanol acetone and ethanol chloroform, and always, again, polyacrylic acid is our main polymer, which we would like to introduce. It's our basic object, I would say, and we want to have on the surface polyacrylic acid. We use different concentrations of polyacrylic acid, starting from 0.25% till 1% concentration, different times, even very long times, for example, 48 hours. We rinsed this material by water a couple times, and we found that we can have in each process, we can receive some number of acidic groups on the surface. This number is dependent of the process conditions. Sometimes it is very long process. It was a very long process, like for example here 48 hours. But it was not necessary to use higher temperatures. So sometimes from the industrial point of view, it is easier to leave something for 2 days than to heat it for 2 hours for 80 degrees, because energy consumption by heating is very high. But leaving it's not for 2 days, it's not, it cannot be a huge problem for industry. Also, different materials, different polymers, which we try to entrap on the surface. It was sodium alginate, like you see here, and we also changing conditions. And finally, we received entrapment degree percent, which can be controlled by weight. So percently, it was more than 3%. And which properties we can change using the surface modification? The easiest way to see the changes in the structure is control the moisture. So the moisture absorption is different, and it is dependent on the number of sodium alginate on the surface. Also, alginic acid was entrapped on the surface, and we looked at the surface using SEM microscopy, and the surface before and after modification is slightly different, but not strongly. At the beginning, it is very regular. After modifications, we can see some irregularities. We used, again, FTIR, ATR spectroscopy to control the structure of the surface. And, of course, next properties which we can change. So the first one is a hydrophilization, because polylactide is hydrophobic, so we can change the structure of the surface to be more hydrophilic. And the next one is electrical properties. For example, static electricity or kinetic electricity. And here you can see that the changes in streaming potential depends on the final layer. For unmodified polylactide, it was minus 846 millivolts. with entrapped alginic acid, it was minus 900. And polyvinyl pyridine, like you see here, was the next layer, because our final expected effect was to have multi-layered structure. Because having multi-layer structure is very comfortable, because we can, controlling the number of the layers, we can have exactly controlling the number of the layers and the type of the layer. We can have exactly such properties which are expected for us, which we need for some aims. So here we start to introduce next layers. What was the mechanism of the introducing of next layers? I will tell you in next slides. But here were introduced the second layer. It was polyvinyl pyridine, as you can see here. Here is a structure of 3 layers, polyacrylic acid again, and polyvinyl pyridine again. We calculated the number of the changes in each gram. It depends on the type of the layer. So these results are showing us that we can control the structure by the number of the layers and the type of the layer. When we finish grafting and entrapment, our industrial colleagues told us that it is too difficult and this is too, too expensive, and they don't want to use such modification. So we were thinking about it. And what we start to do? We start to do just padding. It's a scientific name of very easy process. So what I mean padding? Padding is just introducing the material into some solution. And some things, some polymers or some chemicals from the solution are connected at the beginning using physical attraction to the material. And when the material is dry, they're all still on the surface. And now we have to do something to make the connection or to make insolubility of that padded material. Again, we used polyacrylic acid for padding, and we used different concentrations, and finally, using different concentrations of polyacrylic acid, we received different depositions degree and different hydrophilic properties. So it can be also efficient technique and very easy, but we have to remember that the connection between the external layer and the main material is very weak. It is not so strong as by grafting technique. But sometimes when we are using disposable textiles, it is maybe not necessary to have such strong connection. It may be just weak connection is enough, but this process is very cheap, easy, and we can do it fast. Again, creating multi-layer structure, the first layer is polyacrylic acid with the commercial name carbopol. It is partially a cross-linked polymer, and as a second layer, we introduce here this polymer. I would like to say a couple words about that polymer. We called this polymer with acronym PDAMA. This is dimethyl amino ethyl polymethacrylate. Very interesting polymer, and I am working on this material very long time, and it will be a couple words about that material today later. And it is very interesting because it has tertiary nitrogen here. It is easy to make ammonium salt of this tertiary, to make this quaternary salt. We can do it in easy way. And as you know, the quaternary ammonium salts have very good antimicrobial properties. But as we will see later, here we have antimicrobial properties even at the tertiary salt. again, about the hydrophilic properties. So we are changing the conditions of the padding reactions, of the deposition of both layers, and finally we received material which can be which can have very different hydrophilic properties, different water retention, different moisture absorption, and different deposition degree. Instead of FTIR spectroscopy, here we calculate the increase in acidic groups on the surface. As a second layer, we used also alginic acid, and again controlled the deposition degree and the hydrophilic properties. And here I would like to say, I would like to show you K2S. This is the Kubelka-Munk relationship. The main problem with surface modification always is that you need some advanced techniques to control the surface after modification. So it is not so easy to do very fast. We want to make the fast control. We want to find some method which will be useful for fast control. So we can do it quickly. And we know that there are some dyes which can be used only for dyeing of some special surfaces. For example, maybe it will be here. It will be later. Now, my favorite technique, it was the topic, the topic of my habilitation. It's a layer-by-layer technique. The main idea of the layer-by-layer technique is that we can deposit different oppositely charged polyelectrolytes on the surface, and we are creating the multi-layer structure. The layers are connected to each other using its electrostatic attraction. So one layer must be charged on minus, and the second layer must be charged on plus. This is the basic material. The red one here, this is the first one layer charged, for example, on plus or on minus, maybe on minus. And here, when we take this material into the solution of the other one, polyelectrolyte, we can have electrostatic connection between the red one and the green one. This is easy to do. We do not need to high temperature. We are using here at this technique very diluted solutions at the room temperature. The process takes 20 minutes. So it's the efficiency here is very high, and the changing of the surface properties is really big. And we are, of course, still not changing the mechanical properties of the main material. And we have to know also that it's a kind of nanomodification, because this is the macromolecule, and this macromolecule has its size between 1 till 10 nanometers. So from the one side, it is of course very good to have such thin layers, but on the other hand, it can be not so good, because it is possible probably to use it only for disposable textiles, because it is not, we cannot wash it, for example, because this is too thin, and so strong mechanical interactions as we can have in the washing machine can be too strong for that material. But always the main problem with the application of the layer-by-layer technique is that all chemicals must be electrolytes or polyelectrolytes. So all chemicals, all layers must be charged on plus or on minus. And of course, the modification material should be also charged. It is difficult because we do not have textiles or polymers which are polyelectrolytes. This technique is known since about 20, maybe 25 years. Firstly, it was used for modification of glass, mica, gold, silver, platinum, etc. I found maybe 10 years ago some application of the layer-by-layer technique for one separate cotton fiber, but real modification of textile product was initialized in my unit, in my division. So we were first in the world which applied this technique for modification of the real textile, flat textile material, not a separate fiber. Of course, we started not with polylactide, because polylactide is not so easy material. We started with polypropylene, which is the easiest material. The surface structure is very regular, and the modification should be easy. So we started with polypropylene. The first stage, and this is the main problem with the application of the layer by layer, that we need to find some method to introduce the first layer. And in this, in polypropylene and also polylactide, we use grafting process. So layer by layer deposition itself, like you see here, is very easy, very cheap, and very fast, but always it's a problem with the introduction of the first layer. So we have to use grafting process, so 2 stages process, activation and grafting, both stages at the 80 degrees, as you saw about the grafting. And when we have grafted material, we can deposit polyelectrolyte, polyelectrolytes oppositely charged. So one on plus, second on minus, one on plus, next on minus, but not too much. In the literature, we can find the application of the layer by layer technique, and they used, for example, 100 layers or something, but it is not possible for textiles. For textiles, we can deposit 5, maybe 6. I received here 4 layers. So again, the activation and grafting, 80 degrees here, 80 degrees here, strong concentrations, 20 grams per liter, 52 grams per liter. So it is not a nice process, but it's necessary. Maybe we will find some method, but I will tell you about that later. And this process is also possible to use for polylactide, the grafting process. We saw it previously. And as a charge, as a polyelectrolyte charge on minus, we used my favorite polyacrylic acid, and also polyelectrolytes charged on plus, we use polyallylamine hydrochloride, PDAMA, this polymer, which I tell you that is quite new. Okay. And we received structure, multi-layered structure, unmodified, grafted with 2 layers, 3 layers, 4, and finally with 5 layers. Of course, we have to remember that it must be on minus, on plus, on minus, on plus, on minus. It always must be oppositely charged. And we can see the changes in the structure, the colors. The color visibly is different when we dye this material with methylene blue dye. And the changes in the color we can measure. We have a special apparatus for that, and we can calculate the Kubelka-Munk relationship as you can see here. The unmodified material is probably too bright here to see the differences. The unmodified material, the methylene blue is used for dyeing surfaces which can have acidic character. So this one should be blue. This is unmodified white. After grafting, more blue. With 2 layers should be again white, and it is. With 3 layers should be more blue. It is more blue. And with 4 layers should be again white. And I think it is again white. We can calculate the Kubelka-Munk relationship, and you can see that white is unmodified, blue are with the external acidic layer, and red are with external amine layer. So this is in comparison to this. 1, 3 acidic, O unmodified, 2 and 4 with amine layer, and the area under acidic is much bigger than area under amine, under samples with the last one amine layer. When we calculate this area, we can take the maximum, or maybe we can calculate just area under this relationship. And you can see that this is unmodified, big, with one layer, smaller with 2 layers, and the final amine layer. Again, the final acidic layer, and again amine layer is as a final. As you can see here, and you will probably see in next slides, the differences here will be smaller. So difference between that one is big. Here is smaller, and it looks like this. So it can inform us that we can deposit only couple layers, not too much. Here we used polystyrene sulfonate, not polyacrylic acid, and it is again white, blue. So such relation is possible for different layers, and we also controlled the structure using electrical measurements. For example, the charge, it depends on the number of layers. This is different example here as negatively charged. We use polystyrene sulfonate, and positively it was chitosan, and again there is such relation. When it looks like this, it means that the modification was effective. Here we calculated again Kubel-Kamunck relationships for multi-layer structures, and it is small, bigger, smaller, bigger, smaller, bigger. This relationship can be easy, this modification can be easy controlled using these techniques. So which surface properties we can change using the surface modification? We can change the hydrophilic properties like moisture absorption, water absorption, etc. We can change electrical properties like static electricity, which is a very big problem for many textiles, like comfort of wearing connected with. So we can changing surface, we are changing surface properties. But just by accidentally, I would say, because the thermal analysis is kind of my topic. When I was starting work in the Department of Physical Chemistry of Polymers, it was 14 years ago, we bought at the same time, when I started to work there, we bought a thermoanalyzer. Thermogravimetric analyzer. And I was the first user of that machine. I decomposed it and composed it 2 times. So I know, I have to say that I know a lot about that apparatus, and I like it very much. And every material which I have, I try to test. And so it was no reason to check the thermal properties of these materials. Because the thermal property is a volumetric property. It's not a surface property. But I introduce all samples into the TG analyzer, and the temperature of the composition of unmodified material is 332 degrees. The sample number 1 is sample after grafting. This is PLA with PAA. And here, temperature of the composition is 335 degrees. So it changed about a couple degrees. When I analyzed the sample with 2 layers, the temperature of 50% of the composition were still different. It is still bigger. And again, and finally, final difference between unmodified sample and sample with 5 layers was about 33 degrees. So it is quite big difference. So what is the conclusion from this stage? The conclusion is that using surface modification, we can also change some volumetric properties, not only the surface properties. It was surprising for us, and the explanation, I tried to explain it, but I'm not sure it is good explanations. But the observation is that it is possible also to change the thermal properties of the material. This slide, it was next aim. We are still by the surface modification, and next aim was to have polyaniline layer on the surface. Polyaniline can have different structures, and the color of the structure depends on the conditions. We have leucoemeraldine. This is the polyaniline. Yeah, you can see. This is the green protonated emeraldine, blue protonated pernigranilin, violet pernigranilin base, and again blue emeraldine base. So it depends on the conditions. We have different structure of polyaniline, and finally, the material looks differently, and it is difficult to deposit polyaniline on the surface. But we found a method. This is, I would say, next method. We have our material into the monomer, and we starting polymerization on the surface. This is not polymerization in the some separate glass close to our material, but it is a polymerization, in situ polymerization on the surface. And the effect was very nice. We controlled the structure of the material using standard, even it is possible to see using standard transmission FTIR spectroscopy. FTIR spectroscopy is a basic analytical tool for us. Because the samples have different colors, we can see it also on the UV visible spectroscopy. We can see different maximas connected with different samples. And we used the sample with polyaniline on the surface as a basic for deposition next layers. We use different layers like poly-PO and polyacrylic acid also to deposit on the surface. And here I would like to show you again the TG curves. Here is unmodified materials, and here is a material with deposited polyaniline on the surface. And we can see this is the difference. So it is more than 100 degrees. It is, of course, not one macromolecule layer like it was by layer technique. So we can expect some changes, but this is very big difference. So we can strongly change the thermal stability of our samples using such kind of modification. And it is, I think, something new. And coming back to this polymer PDAMA, which can have, as I said to you, which can have very good bioactive properties, I would like to show you these results are already published last week. So you can find it in the Textile Research Journal. And what was the aim of the modification? We have, at the beginning, we have had polylactide, biodegradable polylactide material. We used padding technique to deposit different amounts of PDMA on the surface. This polymer is water soluble, so it was easy to deposit because of the water solubility, but we would like, we don't want to use a surface with soluble material on the surface. So we deposit different amounts of that polymer. It was about 1%, about 3%, and about 5%. To stabilize the surface, to make a complexation between that polymer and some other chemical, we introduce silver here in amount of 6%, or copper in amount about 4% and 21%. And we collaborate with some different unit from biotechnology faculty, and we would like to change, we would like to control bioactivity of that material. They were different collections of bacteria, different collections of microorganisms from different sources, from example, from tanneries, from museums, from composting plants. So I don't know how they prepare such collections, but they were physically at these places, and they took samples, and in the lab they created a collection of such bacteria, of such microorganisms. And finally, we used our polymers, our samples, to see how it will be work on how it will be reaction of the bacteria on our samples. But firstly, we changed the properties, we controlled the properties of modified materials, because what was the aim of using of that polylactide materials with these layers? We used this as the masks for filtration aims for people. So we had to be sure that we are not changing of the breathing resistance too strongly. So we control it, and we found that we can do it because it is not changing the structure. Of course, always is the same problem, how to control the surface with thin layers. And we use to this aim a special apparatus which can give us information about the elemental structure only on the surface, couple micrometer in depth. And we found that in the samples with PDMA, we can have nitrogen. After deposition of silver, we can have silver. Also, after deposition of copper, we can see this copper in that samples, and only copper. So we can see only the external layer here. Structure was also controlled by SEM microscopy. You can see before modification, this is very regular structure. After modification, after one stage of modification, we can have some irregularity. Here it's completely different. The fibers are connected each other using the modifier, but it is possible because we have a lot of modifiers here. There is not the main slide here. I don't know, it disappears. But I can tell you because I remember what was there. And we saw that using only tertiary nitrogen can kill almost all bacteria, which were collected from museums, tanneries, and different workplaces. So this polymer can be a very efficient tool. And it is even better using this polymer than using usual antibacterial materials like silver or copper. Next step would be the production of the fibers from this polymer. It will be the first possibility the first try in the world to produce the fibers from polyelectrolytes. So, as you see, we are almost finished. I have prepared for you some information materials in English and in the Russian language if you want. You can take in some souvenirs from my university. If you would like to come, for example, for PhD study, to which you are strongly invited. And thank you for coming and thank you for your attention today. Thank you.